Thank you very much. I'm very excited to be here. Can you all hear me well in the back? Do I need a microphone? Okay. All right. I am very excited to be here. I am an assistant professor in the Department of Chemical and Nano Engineering. So we actually just changed our name about a month ago to Chemicals and Nano to kind of be more representative of the programs that we teach in our department. So our department taught the chemical engineering program curriculum, but we never had a department under chemical engineering. So our new department chair, Yufeng, worked really hard to get our department name changed to chemical and nano engineering. So now we can be representative of kind of research and the programs that are being taught in our department. So I joined about 3.5 years ago now. And I thought today I'll talk a little bit about my background, how I got here, and then about the broad goals of my research lab does, and then get into specifically one project that, one story that has been developing in our lab in the past couple of years, which will be actually published next week. So excuse me if my slides are not super polished because these are really new results. All right, so let's start with my background. So I started my education in the Middle East. I was born and raised in Tehran in Iran. And then I immigrated with my family all the way to Canada. And I started off my education in interdisciplinary areas. So my program was actually called Mechatronics Engineering. If you haven't heard of that, it's basically a mix between mechanical engineering, electrical engineering, and computer science and engineering. The idea is to be able to build all the electrical and mechanical components of a robot and to be able to control it. So we took courses from these 3 departments. And I mean, I love Canada and I love everything about my life in Canada, but as soon as I could make my own decisions about immigration, I decided that I would only move to warmer places. So I immigrated to Northern California. And on the flight, I was telling the flight attendant, oh, I'm so excited to move to a warm area. And she told me Berkeley is not warm. So anyways, I was a fool thinking that Berkeley was going to be warm, but still it was warmer than Waterloo in Canada. So I did my PhD again in interdisciplinary areas. I was with Professor Mohammed Morfad, who was between the departments of mechanical engineering and bioengineering. And what I studied there was really looking at how cells respond to mechanical stimuli. And we looked at multi-scale level, looking at proteins, especially proteins of the nuclear membrane and how they respond to mechanical forces. And then for my next move, I decided, please, like, I really want to go somewhere warmer. So I did my postdoc at Stanford, and luckily Palo Alto was much warmer than Berkeley. So at Stanford, I did more applied research in the lab of Professor Bianchao Chui. So she employs some of these mechanobiology some mechanosensitive responses of cells to materials and uses that to develop sensors and materials that can interact or interface with cells and be able to sense different processes within the cells. So for my postdoc, I developed nanoelectronics that could measure electrical signals from electrogenic cells, such as heart muscle cells. And then finally, I was able to move to the warmest and nicest city of all, San Diego. I got a position in the department of, at that time, nano engineering, which was very well suited for my training because, again, I was trained in electrical, mechanical, computer science, but everything that brought my research together was really the fact that it was mostly done at the nanoscale. So we looked at phenomena at the nanoscale, and that's why the nano engineering department really was a good fit for me, in my opinion. So my lab here, we are the Bion lab, bio-nanoelectronics lab, and we're really focused on developing nanoscale electronics and materials that can sense and deliver materials to cells. This is now a couple-year-old picture of my lab. There's a few more people that are added here, but most of the PhD students in my lab are in this picture and some of the master's students. So a lot of the work that I'll be talking about today are done by this wonderful group of people. So let's talk a little bit first about what motivates our lab and what we're actually trying to do. So you might have, as I mentioned, we've developed nanoscale materials and devices for intracellular sensing and delivery. So we develop sensors that can interface with biology. And you might have heard of several such sensors. For example, wearable sensors they interface with the skin. So they need to be designed in a way where they can maybe penetrate skin barriers or be able to sense phenomena on the skin. There are also other sensors like this Utah array you might have heard of, which is an implantable chip that is implanted in the brain and can interface with neurons, brain cells, and be able to stimulate and record from brain cells. So as opposed to these larger scale electronics that interface with biology, we're interested in this area where we're looking to build electronics and materials that interface directly with single cells. So there are, of course, many technologies that are being developed for this single cell sensing. Some are made of synthetic materials, some are made of proteins, different types of material that can interface with cells at the single cell level and sense and deliver materials to cells. Now, why are we interested in single cells? This idea of the cell being the basic unit of life and really enclosing very detailed information about the condition of itself in health and disease. So if you remember this textbook, if you remember from your high school biology, usually cells are represented with this diagram as this entity that encloses its constituents with a plasma membrane. Probably one of the first reasons life began is because cells were able to enclose their constituents within this lipid bilayer to be able to separate themselves from the environment and to be able to do chemical reactions within this enclosed environment. And so this is how cells are able to separate themselves from the exterior. And within this environment is where a lot happens. So we're able to use 3D rendering of eukaryotic cells here and some modeling, not us, other people have done this, to really show the complexity of this intracellular space, all the different components that allow the cell to have its specific functions, whether it's all the material that's enclosed within the nucleus, with different organelles within the cell, or within the cytoplasm of the cell, and then between different cells. And these microscope images are really nice because they, again, allow us to see the complexity within the cell. However, we know that the interior of the cell is not static. And so microscope images show us, these electron microscope images show us some information about the complexity, but they don't give us information about the interactions and the dynamics of the intracellular space. So our ultimate goal is to be able to develop sensors and materials that can interface intimately with cells for long periods and to be able to sense these biochemical activities in the cell. So our sensors would like to have electrochemical sensing capabilities. So that's our motivation for developing these materials. And obviously, if we understand the cellular processes and we're able to manipulate cellular processes, another application for our work is to be able to engineer cells with new capabilities. So if we're able to understand how the cells function, what are the intracellular processes, we can also alter those intracellular processes for our benefit, for therapeutics. And this is already being done, of course, in many areas, for example, immunotherapies, where you engineer your immune cells to, you take your immune cells from a patient, you engineer them to do a specific function, and you implant them back into the patient to go and fight specific diseases. And this is already done for cancer therapy, for example. You take T cells from patients, and you can program them to display some specific proteins on their surface. And then you grow these cells in a dish, and then you implant these back into the patient, and then these are able to recognize and destroy cancer cells. So imagine that we have all these cell types in our body. Imagine if we could engineer cells with new capabilities that could actually live within our system as well and fight diseases. So again, these are the overall motivations for our lab. But to be able to sense the intracellular processes in intracellular space, we have, of course, barriers. Just like if you want to sense from the skin, for example, we have microneedles that can penetrate the first layer and be able to detect from inner layers of the skin, or you have the brain chip implant where you have to open the brain and interface with the tissue. Here we also have for sensing from cells, we have the main barrier, which is the plasma membrane. And if you want to sense from any intracellular organelles, we also have any other membrane-bound organelle that is a barrier to sensing and delivery. So to overcome that, we and others are inspired by nature. So for example, if you look at nature, there's a lot of areas where you see that cells or materials are able to overcome these biological barriers. For example, if you look at the cicada wing, one property that it has, it is self-cleaning. It is self-cleaning, and some recent studies have shown that one of the mechanisms by which it is self-cleaning is its nanostructured surface of these wings. So if you look at the cicada wing under the SCM, you see it's made up of these tiny structures, kind of nanopillar or nanoneedle-like structures. And what they can do, they can actually penetrate the bacteria cell wall as the bacteria adheres on these surfaces and can create ruptures in the wall of these cells, of making them detach from the surface of these bacteria. Other places you see these kind of spiky structures are, of course, on viruses, where these spikes not only allow the cells to penetrate mammalian cells, they increase by increasing surface area, but just by having these spiky structures, they interact differently with cells because of these structures on their surfaces. So we and others are inspired by these structures that are found in nature. And so these nanostructured surfaces like nanoneedle pouches, patches, spiky nanoparticles have been used by us and others for overcoming these biological barriers. So the core of a lot of the work that I'll be talking about today is based on these bio-inspired designs, these freestanding cylindrical structures. So these here are SEM images of these structures. They are basically just attached at the surface here. How we make them is we pattern some circles on the surface of glass, and then we etch down the glass to get these kind of structures. And what we end up with is, it's really just like a glass chip that looks like this. I'll pass it around so you can see. And on the glass chip, you have flat areas, and then you have areas which have these nanostructured surfaces with different geometries, different spacing, different dimensions. So I'll pass that around. And so we can put cells on these and look at how cells interface and interact with these structures. And feel free to stop me and ask questions in the middle of my talk if you have any questions. So what is our approach in my lab? I guess in general is our goal is to build sensors, but we also have to understand how our materials interface with the cells that we're going to sense. So we have this kind of two-tier research approach where a lot of our research focuses on just understanding how cells interface with these nanomaterials. So for example, here's mammalian cells sitting on top of these nanopillar structures. And one of our interests is, okay, what does the cell do? How does it interface with this? What are the impacts of these structures on the cell? And really just fundamental understanding of how cells interface with these nanomaterials. And then we use that information to obviously design our sensors, because our sensors are going to be interfaced with cells at the end of the day. So it's a little bit more complex compared to, for example, designing a sensor that's going to detect oxygen from the air, because now we have this feedback from the cell, and we change the function of the cell as we try to interface with it. So we both of this research happens in parallel. So we engineer nanomaterials and devices. We try to understand how the biology interfaces with our sensors, and these two feed into each other to make better sensors. And then they've actually led to a very interesting biological discoveries as well, by just looking at the interface of cells with these nanomaterials. So what I'll present today mostly will be focused on this area rather than the sensor design. So I'll just talk about some interesting findings about how cells interface with these nanomaterials based on some recent research in our lab. So over the years, because a lot of the focus was on how do we overcome the plasma membrane of the cell to be able to sense the intracellular space, a lot of researchers we're working on looking at the interface, which is the plasma membrane of the cell with these nanopillar structures. So for example, this is a FIB SEM image of a cell interfaced with these materials, the glass nanopillar. And you can see what happens is that the membrane wraps really tightly around these nanopillar structures. And this tight adhesion or tight interaction of the cell with the nanopillars had a broad implication because now, for example, if you're doing electrical sensing, one of the most important parts of being able to sense the electrical activity of cells is to have a very tight seal between the cell and your electrode. And so these structures showed that they have much better seals than just a flat electrode, and they were able to give much higher resolution signals than just a flat electrode. And then another application was if you coat these with some metal, you can actually apply little electric pulses that can open the cell membrane and be able to sense from inside the cell. So there's a lot of work being done at the interface of cells with at the interface of cells at the plasma membrane and the nanomaterial interface. Even us, we studied a lot about how the plasma membrane interacts with nanopillars. We showed that, again, a lot of endocytotic events are activated at the site of the nanopillar with the cell, and this causes the cells to try to basically uptake the pillars, and this is why they had this really tight junction between the cell and the nanopillar. And this is kind of the same mechanisms that viruses use to invade cells basically by activating some endocytotic events at the cell membrane and being able to penetrate the cell. As opposed to these, our current structures are bound to the surface, so they can't be completely uptaken by the cell. You see if the interface is not so tight, they can easily detach even the pillars from the surface. So there was a lot of research done in this area. I didn't even cite all the papers, but there's so much being done at this interface. But what I want to talk about today is about this, this area that kind of started a few years ago that shows that nanomaterials not only deform the cell membrane at the interface of the cell with the material, but they also deform subcellular organelles. So for example, here's an image of really, really sharp pillars that actually are deforming the nucleus of the cell. And this was a study published about 2015, but really after that, there was not much done about, okay, so they deform the nucleus, so what are the consequences of this deformation? And that's really one area that we wanted to study, which is what are the consequences of nuclear deformation by these nanostructured materials? And this research started about 2 years ago in my lab, and it started with this image, which at first gave me a little bit of goosebumps because we've been looking at cells on nanopillars forever. I mean it when I say forever, but we had never looked particularly at the nucleus much. And so what you're seeing here is basically one of these glass chips that I was passing around. And on the top here, like draw a line, the cells are all the same cells on the same chip. The line here is where the nano pattern structures are separated from the flat surface. So the cells up here are on a flat surface, the cells down here are on these surfaces, on the nano pillars. And if you can appreciate what we see here is we are staining for a molecule, which is supposed to be only in the nucleus. So on the top, you see that this molecule is only found in the nucleus of the cell. So the top again is the flat surface, the bottom is the nanopillar surface. And on the top, you see this molecule, not really important what it is, this molecule tagged with GFP is only in the nucleus. And then right when you get to the nanopillars, almost on every cell, you see here that this molecule has breached to the cytoplasm. And it's not left the cell, so it's still in the cell, in the cytosol, but it has left, some of it has left the nucleus. So we thought this was very interesting, and it shows that there's some kind of breach of the nuclear membrane on these nanopillar structures. So this, again, started this area of research in my lab, looking at how nanotopographic materials and just by interfacing cells with nanotopographic materials, you can breach the nuclear membrane of cells. And of course, we wanted to quantify these breaches in some way, or the incidence of these breaches. And for that, I guess the images don't show up so well on the screen, but we basically stayed for this molecule Ku80, which is a DNA repair factor. So it's really hanging out in the nucleus until there's some DNA double strand breaks to fix them, basically. And it should be only in the nucleus. But as you see, again, in nanopillars, in some cells, in several cells, these breaches happen. It doesn't show so well here, but basically the Ku80 leaves the nucleus and goes to the cytosol. And how we quantify this really is just by quantifying the cytoplasmic to nuclear ratio. So how much of this protein is in the cytoplasm versus how much is in the nucleus for all the cells. And to identify cell boundaries, we have an actin stain. To identify nuclear boundaries, we have a DAPI stain. So the DAPI stays in the nucleus. The Ku80 leaves the nucleus for the ruptured cells. And this is the chart I'll be showing a lot. So by quantifying the cytoplasmic to nuclear ratio of this protein, which is supposed to be in the nucleus, we can identify cells that are ruptured. And we did some analysis to identify a cutoff for where the cells, what cytoplasmic ratios have Ku80 correspond to the cells being ruptured, and that was about 0.16. So you can appreciate here that on the flat, the ratios are much smaller, meaning there's not much Ku80 in the cytosol, and on the nanopillars, there's a lot of Ku80 in the cytosol. We tested different cell types. Of course, different cells have different nuclear mechanics, different properties. So we want to see if this phenomenon occurred only in one cell type, which was this epithelial U2OS cell, or does it happen in other cell types? And the few cells that we did test, we saw ruptures in all of them. In these cardiomyocytes, we did not see it in wild type iPSCs, but we did see it in lamin silent iPSCs. So that's an interesting thing here, where none of the wild type cardiomyocytes show any rupture on our structures. So again, this is just quantified by looking at how much of this Ku80 goes into the cytoplasm. So the red areas are the Ku80 in the cytoplasm. You can see a lot of red areas on the bottom half, which is the ruptured or breached cells, and on the top, you don't see much of this breach. The next thing we asked is whether these breaches are actually permanent, because the cells, I mean, there's years of studies of putting cells on these nanostructures. So proliferate, they have up to like the knowledge of most people in this field, they function properly and they survive on these surfaces. So we asked whether these breaches that are happening in the nuclear membrane are actually permanent. And the answer was no. So they actually are able to repair themselves over time. And how do we see that? So again, here's the nucleus of a cell. It's not breached because we don't see the cytoplasm. As soon as it breaches, this Ku80 gets into the cytosol. And about 30 minutes later, we see that again, it's not shown very well on this screen. The contrast is not great, but you still see some in the cytosol. And then by 60 minutes, it all goes back into the nucleus. So this suggests that that breaches are able to repair. So you can see here about this time point is when the cell starts breaching, but the cytoplasmic ratios of this molecule go really high. And then over time, they repair and go back to their normal levels. So the summary so far is we learned that engineered nanotopographies, these materials with some nanostructured topographies on their surface, can actually transiently open the nuclear membrane. They can transiently breach this barrier of the nuclear membrane. And I'm trained as an engineer, but I'm a scientist at heart, because when I see something like this, before I think, oh, what can I use this for? I think, oh, how does this happen? How is this happening? So I think about the mechanisms a lot. Obviously, reviewers, all they ask is, oh, okay, what can I use this for? What can I use this for? We'll get to there, but we also have to understand the mechanisms. So we are doing this in parallel. I'll talk a little bit about both of these. So this is new research in my lab. We're working on both. We haven't answered all the questions, but we have, let me go to the next slide first. Here. We are looking into factors that increase or decrease the incidence of these breaches So first we looked at the material factors and the cell material interaction factors. So we looked at whether the time of interaction between the cells and nanomaterial impacts these breaches. We saw that with when you put the cells on these surfaces, in the first hour, you don't see much breach, so 6.5% of the cells show breach. In 5 hours, there's a significant increase in the breach incidence and not much more after 8 hours. So between 1 and 5 hours is where most of the breaches happen on this specific nanopillar structure. And we do confocal imaging to actually see the curvature and the indentation that the cells and that the nanopillars impose on the nucleus of the cell. So here is a confocal image. Again, imagine like a slice of the cell looking at it from the side. And this is the nucleus of the cell. So this is a lamin AC stain, I believe. And what it's showing is basically the nuclear membrane, very close to the nuclear membrane. And from this, we can actually see the deformation of the nucleus from the nanopillars. And we do some quantification of the indentation and the curvature of the nucleus at those points. And what we show is that one thing that is changing between 1 and 5 hours is this indentation depth of the nucleus with the nanopillars. Again, because the cell starts wrapping around these nanopillars and is indented by the nanopillars. Another factor that's changing is the curvature of the cells of the membrane on the nanopillars from 1 to 5 hours. So the time or duration of interaction of the cells with the nanopillars is one factor that impacts these regions. The other question we ask is, how does the geometry of these nanopillars impact these breaches? So on the flat surface, again, we see no breaches at all. When we have larger pillars, and these are up to 700 nanometers, I think, in diameter, and 2 to 3 microns in height, we see about 12% breaches on these structures. But once we shrink these structures, so what we do is to keep all the other parameters constant, we just take these glass pillars and we use some very diluted HF to etch these pillars down into smaller scales. So these are the smaller pillars. And when we made them really small, we saw up to 78% breaches on these structures. And of course, we asked how do the indentation and curvature parameters look on this? And you can see that although the indentation depth is much smaller on the small pillars, because of course they're shorter, the breach incidences are much higher, suggesting that the indentation depth is not very important in this case. We did see a small change in the curvature from the small pillars versus the large pillars. So a sharper curvature is what we're seeing on the smaller pillars. We looked at some of the properties of the material that can impact the incidence of these breaches. We asked, what are some properties of the cells that are impacting these kind of breaches? Sorry. And we looked into some specific proteins in the cell that are known to be responsible for maintaining the structural integrity of the nuclear membrane. So one of those is this lamin AC. So lamin AC is one of the main contributors to the nuclear membrane integrity. And decreased levels of lamin AC have shown to lower the nuclear stiffness. So we hypothesized that if the nuclear stiffness is lower, we can deform it more easily with our nanopillars. And therefore, we would see an increase in the incidence of these ruptures. And it seems like that was the case when we silenced the lamin A gene, which is encodes for lamin AC. We saw significant increase in the incidence of ruptures on our nanopillars. And we also looked at the role of actin polymerization and contraction. As we know, actin dynamics are very important for maintaining cellular and nuclear architecture. We asked whether this maintenance of cellular and nuclear architecture is important for these nuclear breaches. And we used 2 drugs to impact actin myosin contraction and actin polymerization. You can see that with the control where we had DMSO, which is the carrier of the drug. You see about 44% rupture in this case. When we do the actomyosin contraction inhibition, it lowers it to 18% rupture incidence, so fewer cells show this breach of the nuclear membrane. And then when we inhibit active polymerization, we see only 2% of rupture. So this suggests that the contraction alone is not explaining all of the incidence of breach, but active polymerization seems to be very important in the cells exhibiting these breaches. So to summarize so far, what we've learned is that the curvature induced by nanotopography on the nuclear membrane can cause these breaches. We don't know exactly where these breaches are, but in the schematic suggests that this curvature is what causes the breach, and that's where the breaches are. We showed that smaller nanopillars can induce higher rates of these breaches on the nuclear membrane. And we showed that many cell types exhibit this breach in their nuclear membrane when they interact with nanomaterials. There's so many questions remaining. We're collaborating with many people, including Dr. Randamani, to understand what are the mechanisms to model how the nuclear membrane is rupturing in these cases. But we are also thinking about the applications. And I really think there are many applications for this finding. And the first one is to be able to deliver things to the nucleus itself. So in the field of delivery, of course, many people focus on how to overcome the cytoplasmic barrier, how to overcome the plasma membrane to get materials into the cell. Of course, that's important if you're delivering RNA, because it needs to get access to the cytosol. If you're delivering some specific small molecule or drug, that's all it needs to get into, which is the cytosol. But if you're delivering something like a gene, the ultimate goal for that is to get into the nucleus, because that's where it's going to be transcribed and then moving to the cytoplasm to be translated. So many material that you need to deliver to the cell ultimately need to get into the nucleus. So getting into the cytosol, there's many cool, exciting research that is being done in this area, and many materials like lipid nanoparticles, different viral vectors to get material like DNA into the cytosol. But then how does it get into the nucleus? And currently there are two major methods for nuclear delivery. One is through mitosis, and this is the most common method by which when you deliver something to the nucleus, it gets into, deliver something to the cytoplasm, it gets into the nucleus. And that's when 24 to 48 hours after you deliver your material, the cell will divide. And during this division, the nuclear membrane breaks down, and that's where your cargo gets into the nucleus. Of course, there are many drawbacks with this method. It's the cargo, whatever it is, it's waiting in the cytoplasm for a very long time. Also, if the cells are not dividing, you can't use this method to get your cargo into the nucleus. So this definitely decreases efficiency of delivery, because the cargo, especially if it's, for example, single-stranded DNA, it gets degraded really quickly in the cytosol. So getting it rapidly into the nucleus is a challenge. But this is the main method by which most cargo gets into the nucleus. The other one is to transport it across a nuclear pore complex. So as many of you may know, the nucleus actually has its own large macromolecular complexes that control the import and export of material into the nucleus. So in order to get through this pore, you have to either be a very small molecule to just diffuse through it, or if you're larger, you have to have these you have to be actively transported across the pore. And to be actively transported, you can attach these signals to your cargo, whatever material you're trying to transfer, to be able to attach to these important molecules. So these important molecules basically allow you to be actively transported across the nuclear pore. So some people put these signals on their cargo, and so this cargo is able to be transferred across the nuclear pore complex. Again, there are several limitations with this. You have to add more, obviously, signals onto your cargo to get it through. It makes it bigger. And then also there's a huge size limit of what can go through the nuclear pore complex. And so, and then also this process is also quite slow. It's not that fast. So there are several limitations with current methods of getting material into the nucleus. And so our proposal is to use this nanotopography induced breach to move cargo or whatever material it needs to go from the cytosol into the nucleus. And we recently filed a provisional patent for this idea. And I'm showing very preliminary unpublished data here. So again, our proposal is that the cells on the nanopillar do not need this mitosis step. They don't need to wait until the cells divide to be able to let the cargo get into the nucleus. So what I'm showing here on the top is a time-lapse video of cells that are transfected with this plasmid. This plasmid encodes for, it's a gene that encodes for just GFP, NLS GFP, which is supposed to be found in the nucleus. So as soon as this plasmid is delivered to the nucleus, you should see a signal basically in the nucleus. So here, this is the first frame where the signal shows up, and you can see as soon as it shows up, there's two cells next to each other like this, suggesting that this was a mitosis event where it caused this gene to be expressed. So again, the cell is dividing. As soon as it starts dividing, you're able to see the signal. The frames before this don't show anything. So as soon as the cell starts dividing, you see the signal, which is again what happens in most cells where you have mitotic delivery, meaning that you deliver your cargo to the cytosol, and then how it gets into the nucleus is when it goes through mitosis, when it divides. And number of transfected cells on flat and nanopillar are through this method are pretty much the same. Again, these are very preliminary data. So on both nanopillar and flat, you see this mitotic delivery is when the cells divide, you get this expression of your plasmid. But in many cases, on nanopillar substrates, the same delivery method to the cytosol, but now we see many cells that don't need to go through division. So it's a single cell, it expresses on its own without going through division. And we see this only on the nanopillars. We don't see this on the flat. So we think this is very exciting. We can deliver potentially faster to the nucleus, which could potentially lead to higher efficiencies. We can deliver to cells that don't divide, because that's a huge limitation of cells that don't divide. We can't get the cargo into the nucleus. So we are working on proving this for different cell types, especially stem cells that are hard to transfect and again, very relevant to clinical applications. So just to summarize, we take this interdisciplinary approach in our lab. Our goal is to build devices and materials that interface with cells to be able to detect intracellular processes for prolonged periods. So we want our materials to be able to interface with cells over long periods. But to do that, we do a lot of fundamental studies about how materials interact with cells. And through these fundamental studies, we've really made some interesting discoveries that then inform new devices that can, with new capabilities to manipulate and sense cells. And we have a lot of ongoing collaboration. Of course, all this interdisciplinary work would not be possible with a single lab. So the work, at least that I presented today, was a collaboration between Joe's lab and Colleen's lab. Sorry, this is mixed up. Lingyang Shi's lab, and yeah, those were the people that were involved in this specific work. But we're working with all these people at UCSD and at other universities to develop our sensors and materials. And then I want to highlight two important collaborations that we have right now related to this project. One is with Lingyang Shi from bioengineering to look at how the lipid dynamics, lipid metabolism of the nuclear membrane versus the versus the plasma membrane impact these kind of behaviors that we're seeing, because we're indenting both the plasma membrane and the nuclear membrane, but the nuclear membrane is the only one that exhibits this phenomenon. So we're very interested to see some of the lipid, the role of lipids in this phenomenon. And then, of course, with Dr. Rangamani's lab, we're trying to model this behavior. So what is happening at the material cell interface, what are the signaling pathways that allow, we see some specific molecules being translocated to the nucleus during these breaches other than the one I showed. So we're trying to see how exactly the signaling happens from the extracellular space all the way to the nucleus to get these molecules to be expressed inside the nucleus. So, and to be able to just model this whole process through biophysical models. And then with that, I'd just like to thank my team again. I'm lucky to have a group of very brilliant and motivated students who did this work. I think all these people were on the paper. Ryan was the one who found that first image where the breach happened. He was a master's student. I told him to stay. It's really exciting, but he needed to make money. I know students think that way. He needed to make money, so he's now at Atomica. And then Ali took on, and he's the main lead on this project. He's going to graduate next year. And then everyone else came on, helps us with a lot of the computational work, how to automate all the analysis of the nuclear cytoplasmic ratios and determining the cell boundaries. Shivani and Divya did a lot of the nanofabrication. They're the people who do the nanofabrication in our lab. And Leia really helped with manuscript revisions and just, making the lab function as a whole. And with that, I'd like to thank you and take any questions you may have.
Oh, you're good. So when you're showing the different delivery and how the mitotic delivery was similar on the nanopillars versus on the flat surface, how do you specify the mode of delivery if both of them are on the nanopillars?
Yes, sure. So the main thing we do here, so we have both this channel, which is we're imaging two channels. One is just this GFP channel, and then we're doing bright field imaging as well. The way that we detect it right now is if two cells show up next to each other during expression. So as soon as the GFP signal is seen, do we see one single cell, or do we have two cells show up together? That's how we're detecting.
You don't there's no specific way to say if you want it to be a mitotic entry versus non-mitotic entry.
So this is basically we're imaging the cells in the same way on nanopillars and flat. Sometimes we even have them in the same view.
Yeah.
So and then we're detecting whether it's a mitotic delivery or not, right? So here we see both of them have enough mitotic delivery because when a cell divides, this will happen. The plasmid will get it.
I realize you're not heading this direction, but you're reminding me a lot of a discussion about 20 years ago about why asbestos was so toxic. And I guess the question is, do you have any insights into what makes some nanoparticles more toxic than others in terms of health
I think
effects?
that's a very good point. And right now we work with the glass surfaces that you saw, so surface attached nanostructures, but I show the nanoparticles a lot because I do want to emphasize that there's nothing special about these structures being bound to the surface. This is just easier for us to study the interaction because the cell doesn't take up the pillars. But if you put these structures on nanoparticles, then you can potentially do in vivo, you can get the cells to interface with them and indent them. So I would completely think that the surface topography of some nanoparticles can impact the response of the cell. So we even see this in our studies. Sometimes the nuclear membrane breaches, but it doesn't repair, and the cell completely goes through apoptosis or goes through senescence, and that's an area that we are trying to write grants for. But definitely, these particles, these structures, if they're indenting the nucleus, they're rupturing the nucleus. I think if they're too large ruptures, they would destroy the cell.
I heard someone speak just yesterday about molecular weights and transport into the nucleus because they were trying to get oxidants into the nucleus. And I wonder.
I would love to talk to them.
Yes, I was going to write you and then an email, but I wonder if in your studies, especially with Ali's paper that's coming out next week, you'd looked at the distribution of the nuclear core complex. Is that something one can do?
Yes, absolutely. We want to look. That's a very you read my mind. We want to look at so we had the Lannan image, remember? Yeah. I want to do that exact same thing, but with all these other proteins, like the nuclear pore complex. So Ali's bought the stain for that. Not 153, I think. but just look at the distribution. Does that change? And I think it would, because it's such a huge complex that curvature will impact it. I'm 100% sure.
Okay.
So what is this nanoscale curvature? How is it changing the overall structure of the nuclear membrane? How is it changing the nuclear pore complex composition, lemon A, lemon B, all the structural components at that place, and lipids.
Yes,
Yeah.
I was going to ask that question. So
Yes.
it's really.
Yeah, but I think one impact of this would be delivering cargo that is potentially too large to go through the nuclear pore complex, and too fragile to wait until mitosis.
Well, when you form these nuclear ruptures, you show the Ku-80 coming out.
Yeah.
But I also wonder what else goes in, right? I mean,
Yeah.
you can imagine, but you can have like a 4 kDa or 8 kDa molecule.
Exactly. So we
go in.
That's a very good point. And we are trying that. It's just getting it into the cell first is a little bit tricky of how to control it being in the cell and then going into the nucleus. Yes.
Long-term cellular survival after this rapid gene delivery?
Yes.
These are very excellent questions, and I wish I had like 10 more students that could answer all these questions.
But absolutely, we want to look at different cell types because each of them will have a different mechanism. Like neurons don't have as much like endocytotic processes, so it will be very different how they interface with these structures. We have not tried neurons. That's the first answer, and I would love to try neurons because we work a lot with neurons, so we just need to have someone to try it. And then your second question, exactly what we're trying to study, because as I mentioned, some of the cells, a percentage of the cells don't survive this breach. So I think that's partly due to the size of the breach. So we really want to understand, like, is there a way to quantify the size of these breaches and then correlate that with whether the cell is able to repair, how long it takes for the cell to repair, because if we're using this for delivery, these are all important questions to answer. But for sure, right now, on some of the cells, we see them, they go through apoptosis immediately after the bleach. Yeah, a percentage. So we would love to study and understand it. And that has its own applications because eventually, if you think of being able to kill the cell with this particle just by indenting its nucleus, I think there's some applications in cancer therapies for that.
I think previous talking approach so more on the design side. And in terms of the nanofillers, you have a certain way of manufacturing these etching, right? Are there certain geometries that you would be interested in pursuing that you can't currently do with the manufacturing method, i.e. some that you may see in biology, like those spikes are pretty bulbous on the end and they get narrow as you get closer to the substrate. Are there certain geometries that you'd be interested in producing that you think would be promising just in manufacturing or technology right now producing that?
Yeah, so first of all, I mean, I would just want to find cheaper ways to make these right now because I don't know if any of you work in the Nano 3 facility. It's very expensive. So we spend a lot of money just making these structures. So I would want to find easier ways to manufacture them for sure. With the etching technique, there are many techniques to make different shapes. With the etching technique, yes, we are limited by the shape of the structures that we can get. We can change the etching parameters, the gases that we use for etching to get tapered versus straight walls. And that's what we're doing right now. But yeah, if you have any ideas or any specific manufacturing techniques that are in your mind, yeah, we'd be happy to talk.
I wish to say thank you to Dr. Jahed for speaking. Next, actually, we will have Kyle Stark, a trainee here. Kyle is a PhD student in the Mechanical and Aerospace department. He has a BSc in Mechanical and Aerospace Engineering from Rutgers. And before starting his PhD, Kyle worked at SpaceX on the Dragon capsule's exterior structure. Now he researches biophysical models on cellular morphogenesis and competitive drug models.
Yes.
My eyes were like, oh.
That's good.
We also wanted to give our speakers, particularly Dr. Jahed, a certificate for coming and speaking. So, sure. Thank you so much.